Muy buenos días, bienvenidas y bienvenidos a todos a esta edición de Conversus Talks. Yo soy Ana Cristina Olvera, coordinadora de la agencia informativa Conversus, de la Dirección de Difusión de Ciencia y Tecnología de aquí del Instituto Politécnico Nacional. Y Conversus Talks es una serie de charlas presenciales con personas clave de la ciencia y de la divulgación de la ciencia, en las que intentamos acercar a los seres humanos, las personas detrás de la ciencia, a la mayor cantidad de público en general. Y hoy tenemos el grandísimo honor de que nos acompañe el doctor Venki Ramakrishnan, Premio Nobel de Química 2009, por su contribución a la descripción de la estructura del ribosoma, que es la parte de nuestras células que crea las proteínas por las que vivimos. Pero él nos va a explicar mucho mejor de qué se trata. Así que bienvenidas y bienvenidos todos, muchas gracias por acompañarnos. Bienvenidas y bienvenidos también a las personas que nos están viendo a través de la transmisión en línea. Vamos a presentarles un breve video de nuestro invitado de hoy.
En lo más profundo de cada célula hay un universo diminuto, complejo, en movimiento constante. Ahí, donde la materia se organiza para dar origen a la vida, ocurre uno de los procesos más asombrosos de la biología, la creación de proteínas. Desde su infancia en la India, Venki Ramakrishnan soñaba con entender cómo funciona la vida. Su curiosidad lo llevó a cruzar continentes y disciplinas hasta convertirse en uno de los científicos más influyentes de nuestro tiempo. Durante décadas, dedicó su trabajo a un objetivo tan simple como monumental: descifrar la estructura del ribosoma, esa maquinaria microscópica responsable de fabricar las proteínas que nos mantienen vivos. El descubrimiento cambió la biología moderna. Por primera vez, la ciencia podía observar cómo el ADN se transformaba en acción, cómo la información se convertía en vida. En 2009, ese trabajo fue reconocido con el Premio Nobel de Química, pero para Venki, el verdadero premio no fue la medalla, sino haber ampliado los límites del conocimiento humano. Hoy, su curiosidad sigue intacta. En su más reciente libro, ¿Por qué nos morimos? La nueva ciencia del envejecimiento y la búsqueda de la inmortalidad, explora el proceso biológico del envejecimiento, las razones evolutivas de la muerte y la belleza que hay en aceptarla como parte de la vida. Entre la ciencia y la filosofía, Venki nos invita a mirar el cuerpo humano no solo como un conjunto de mecanismos, sino como una historia en constante transformación. Hoy comparte con nosotros su viaje por la ciencia, la curiosidad y la vida. Bienvenidos a Conversus Talks, el Nobel que descifró el centro de la vida y el código de la muerte, con el doctor Venki Ramakrishna.
Professor Venki, welcome to IPM. Thank you very much for being with us. We are truly honored to have you here, and we are eager to listen to your experiences. First of all, well, I want to tell everyone, Professor Venki has been here for over a week, a little bit, and he has visited Guadalajara, Feria del Libro. He has been to Puebla. How has been your experience in Mexico so far, Professor?
Very nice, and the weather is a nice contrast to what is happening in Cambridge or in North America at the moment.
And also, what about the weather? Are you glad to be here now?
Yes, that's what I mean, it's a nice contrast.
Please, let's start with...
Ahora hace mucho frío en Cambridge, así que estaba disfrutando mi visita en México.
It's nice to have sunlight and wear light clothes. So to start, Professor, could you explain for the audience that is also tuning through the broadcast, what does the ribosome mean for you?
So, all cells, all life requires proteins to function. So proteins, for example, carry out most of the thousands of functions that are required for life. They also give us form. For example, our skin, our muscles, they're all made of proteins. But proteins also are responsible, for example, for carrying oxygen in our blood to our tissues. They're responsible for sight, because a protein detects light in our eyes. Proteins are also antibodies, so when we want to fight off infection, we make proteins, which are called antibodies. Proteins digest food, so they're responsible for being able to use nutrients from this outside to convert them. And proteins are also involved in making everything in the cell is made by protein enzymes. But how proteins are made was a big mystery. And then when the structure of DNA came out and people discovered the genetic code, they knew that DNA contained information, instructions on how to make a protein. And DNA is copied to mRNA. The gene of, DNA has thousands of genes, and each gene is copied to another molecule called mRNA, which is like DNA, but except it has only one strand. DNA is a double helix. And no one had heard of mRNA until 5 years ago, because it's only molecular biologists who had heard of mRNA. But of course now everybody has received mRNA in their arms because of the COVID vaccine. What that mRNA is instructions on how to make the COVID spike protein, the coronavirus spike protein. But like that, there are thousands of mRNAs. Each one is coding for its own gene, and which makes its own protein for a particular protein. And in every case, the ribosome, which is a very large molecular machine, it's made up of many hundreds of thousands of atoms, maybe 500,000 atoms for the human ribosome. That molecular machine finds the mRNA and starts reading the instructions on the mRNA to make the protein. And that's how every one of your proteins is made. In fact, while I've been speaking, your cells have been making hundreds of proteins in every one of your cells.
So I know you mostly concentrated in one part of the ribosome at first.
Yes, but that was only a small phase. Very quickly, you can only, the ribosome consists of two parts. One is the part that binds to the mRNA and reads the genetic code. The other part is what joins amino acids together to make the protein. The two parts have to work together. So initially, there was a race where a group at Yale worked on the large subunit, which is involved in joining the amino acids to build the protein. And I worked on the small subunit, which is reading the mRNA and reading the genetic code. But that phase only lasted for 1 or 2 years. But for the next 20 years, we've been working on the whole ribosome and trying to figure out how it knows where to start, how it moves, how it attaches tRNAs to it, how the amino acids are brought to it, and then how the process terminates at the end. So really, the vast majority of my life has not been on the small subunit, but on the different parts, different phases of the whole ribosome.
How does it feel to be working in an investigation that could potentially win a Nobel Prize? Because it's not all about science.
At that time, you're not thinking about prize. Scientists don't, unless you're a little bit crazy, you don't think about the prize when you're doing science. So I think what happens is when you're very young, like a student, you think, oh, I will become a scientist and one day I will win the Nobel Prize. Okay, but that's a kind of immaturity. Once you start working in science, you know that the Nobel Prize is not a realistic goal. It's like a lottery, And so you become interested in something and then you work on it. Then, if you're doing something really important, you're excited by the problem. You don't think about the prize, you're thinking about solving the problem. But once the problem is solved and you have done something really important, even if you don't think about the prize, other people tell you when you go and give talks or lectures, they say, oh, one day this could win a Nobel Prize. So it kind of affects your psychology.
How did it affect you? How did it affect you?
I think, it affected you because then you think, you see the Nobel Prize, first of all, it has to choose your topic. There are many topics in science, and there's only one prize every year. So they have to, you don't know if they're going to choose your topic. And I felt that the ribosome was very important because it's fundamental to biology. So maybe I would not have been surprised if the ribosome would get the Nobel Prize someday, but I didn't know when it would happen. But the Nobel Prize is only given to at most 3 people. So then, what happens is you think, oh, will I be one of the 3 or will that other guy get it? And so this also affects your psychology. This affects every important scientist who's done very important work. They suffer from this disease called pre-Nobelitis. Okay? And every October they develop a fever, because they're waiting for this phone call, and then it doesn't come. Then they're unhappy for a few months. Then again they become, optimistic the next October. And this keeps on going, and some people, they die, without, getting the Nobel Prize. And, so I think this is a problem with prizes, because science is not actually, it's not a sporting competition like the Olympics, where you have a 100-meter race. I remember in Mexico City, 1968, all the records were broken, for 100 meters, for long jump, because of the high altitude, there was no air resistance. Anyway, but it's not like science is not like a 100-meter race where you can very clearly see 1, 2, 3, these people came first, second, and third. Because in science, people contribute in many different ways. And who has done the most important work? If you have 5 different juries, they may have different answers, so it's not clear that always, it's not always clear. Sometimes it's clear. But nowadays what happens is there's a lot of communication in science very quickly. So the ideas are followed by many different people. So it's sometimes hard to find out who are the people who really did the work.
How do you think the Nobel Prize has changed from when you won the Nobel Prize to today, and how will it change for the future?
You mean the prize?
The prize.
The prize.
The prize.
I think between when I won it and now, it has not changed that much, okay? But I think in 1901, when the first Nobel Prize was started, first of all, science was a much smaller enterprise. There were far fewer scientists. Secondly, communication between scientists was very slow. There's no like internet, and people didn't travel as much. Maybe they would go to a conference once every 5 years, okay? And so it was easy to see who had done the really big breakthrough. Because you knew nothing, and suddenly after 5 years you would see this person had really just made this big discovery, okay? So there was not as much difficulty in those days. Now science is a very big enterprise, and science is also highly communicated now because of the internet and emails, and people go to conferences. They go to many conferences a year instead of one every 5 years. And so ideas spread very quickly, and lots of people work on important problems. And so that makes the job of the Nobel Committee more difficult.
How was to receive that call? Please tell us about that.
Well, you know, it's very funny because the public only thinks about the Nobel Prize, but there are many prizes that are given before the Nobel Prize, okay? And most of those prizes went to other people for the ribosome, not to me, okay? And so I felt that my chances were not good. And then, you know, but I knew that I was being considered because from 2000 to 2004, every year I would be invited to Sweden for some conference, okay? And often there would be people from the Nobel Committee in the audience, you know, so it was like an audition, you know, like an audition for the prize. So anyway, we'd give our lecture and pretend we're not aware of this. But at one of the last such meetings, where, by the way, many people spoke who later won the Nobel Prize at this meeting, you know. So clearly they had invited people who they thought were good candidates. And at the end, there's a banquet on the last evening. And at this banquet, there was a ribosome scientist from Sweden who didn't agree with me about some aspect of my work, you know, about the accuracy of the ribosome. And he came and started fighting with me. And, you know, so we had this big argument, and then, in fact, it got so bad that his colleague had to come and kind of separate us, you know. And then I went home, and then after a few months I found he was appointed to the Nobel Committee in Chemistry. And so I thought, oh, I'm never going to get the Nobel Prize, because I know this guy doesn't agree with me. And then after that I stopped, you know, if I got an invitation from Sweden, I said, no, I'm too busy. You know, because I thought, why should I go there? I'm not going to get the prize and I just look like a beggar, you know. And so, anyway, it was very funny that after 5 years, I suddenly got this phone call, so I was very surprised. But it shows this guy was extremely honest, you know. It means, most people, you know, very few people would have had his integrity, because this guy, even though he disagreed with me about this one area, he was willing to put that aside and say, okay, but if you look at the whole field, you know, this guy deserves it. So he was very, very honest about it, you know.
So you talked about pre-Nobelitis. We know everyone gets the fever, but you also talk about post-Nobelitis.
Oh, that's even more common. Well, no, I would say it's as common. What happens is that after you win the Nobel Prize, you see, the Nobel Prize is given for an important discovery, okay, or invention. It is not given because you're a brilliant scientist, okay, or that you know everything. In fact, there are many people who won the Nobel Prize who just were lucky, you know, they did something and discovered something by accident that turned out to be extremely important. Even the very first Nobel Prize, superconductivity, he was not imagining that there would be some phenomenon like superconductivity, he won the first Nobel Prize, just by accident, almost, okay? So, and then there are other people who happen to be in a good place, with lots of resources, and they have a problem, and they work hard, and and they make some important advances and contribution. And then there are other people who are just really brilliant, and they just know what to think. And there's a saying in Shakespeare, which is, some are born great, some achieve greatness, and some have greatness thrust upon them. That means, they're not great, but somehow had greatness given to them. And so it's the same with Nobel Prizes. So what happens is, after you win the Nobel Prize, the public doesn't understand that. They think that, oh, you won a Nobel Prize, you must be like Einstein or, something, because Einstein won a Nobel Prize. So, they think you're some sort of genius, okay? And not only you're a genius, but you know everything about every field. Suddenly they ask me about climate change or, things like this. So, and then many Nobel laureates, they have not done any important work for 20 years, okay? Because the work they did was 20 years old and then they get the Nobel Prize. So for 20 years they've done nothing, not nothing, but maybe not very important work. And then suddenly they get the Nobel Prize when they're in their 70s or so, and they, nobody has paid any attention to them. And suddenly they're in the limelight, the press is interviewing them, they get invited everywhere, here and there. They're practically spending all their lives in an airport after that time. And then they start taking questions about all sorts of things. And because everybody takes them seriously, they start believing themselves to be, very wise or stuff. So that's a disease called post-Nobelitis.
So for you, how has it been life after the Nobel Prize?
Well, for the first few years, I really just kept focusing on my science. I was not that old. I was 57. So I was somewhat younger than many Nobel laureates, and especially these days, because, the queue has become longer and longer. So people have to wait longer and longer, and they often get their Nobel Prize relatively late in life. And so I still had an active lab, and I kept doing science, and in fact the next 5 years we published many more papers than in the 5 years before the Nobel Prize. But the thing that changed for me is I became elected president of the Royal Society, which is like being the president of the National Academy of Sciences. And that is a very public position. You have to talk to the public, to the press, to the government, to other national academies, and also you have to talk to scientists from all different fields, and you meet people also not from sciences, but from many different areas. So you become more of a public figure. That, I think, was very different. And I would not have been elected if I didn't have the Nobel Prize. And so that was a big change.
What advice would you give students and young professors about not just having a recipe to win prizes, but also enjoying their research and also navigating the politics of science itself?
What do you mean by the politics of science?
I mean, because sometimes you do need politics and social relationships for your investigations to advance and get funded and get...
Yeah, I would like to think that's not... I think you're right on the whole, but I do know many scientists who are complete introverts who have nevertheless succeeded in science. In fact, a couple of them are friends of mine. And they are extremely shy and not very political at all. So I don't think it's necessary, but I agree that if you're very social, it does help. I think one should probably, the best way to do science is really go into a field that you find interesting and pick a problem that you think is both interesting to you and is kind of important to the field, and start working on that. And even if you make very small steps, it's okay because you're contributing to an important problem and you're interested in it. I think that's the way to do science, to enjoy the process. Otherwise, you'll be sort of frustrated because the act of science can be very tedious. Every day you come, you have to do experiments, it's hard work, sometimes it takes long hours, and you have to work late at night sometimes. So I think you have to be interested in what you're doing.
You also didn't bother about changing fields or learning new skills to solve the problem. If an IPN student told you, I want to change field, but I'm afraid of what could happen with all what I've built in this field.
Yeah, I wouldn't recommend changing fields if you're happy in your field, because it's a lot of work to change fields. I think the reason I chose it is the times I changed things was because I had no alternative. I studied physics as an undergraduate and also as a graduate student, but when I started doing research, I found it very difficult and not interesting. So both were problems. And so if I stayed in physics, I would have become a failure. I would have been frustrated. I might have, even if I had succeeded, I would have not been very good at it. I would have done some boring work and published a bunch of boring papers. And, I'm afraid a lot of science is like that, And so, in fact, there's a joke that many papers are not read by anybody. They're only read by the author and the two reviewers, So I didn't want to be like that, And so I switched and became a graduate student again. After a PhD, I went back to graduate school in biology in San Diego. And but that was almost out of necessity, If I had been doing very interesting physics and and been making good progress, I would not have switched.
So right now, students and researchers and professors, they have a big component in their lives that was not there when you did all your research. We were talking about it earlier. It's AI. So what advice would you give scientists, also professors, about what AI can do, and how do you think, most of the recent Nobel Prizes are given to people that use AI for their investigation, so how is this going to change investigation?
Yeah, so they actually, I think they were inventors of AI. I would say they developed AI for other people to use, like this AlphaFold in my field of structural biology is an AI-based program that allows you to calculate the structure of a macromolecule, whereas before you had to determine it experimentally. And of course, it's not 100% reliable. With any AI, it can do what are called hallucinations. It can give you completely the wrong answer sometimes. But nevertheless, it's been extremely successful, and that's why it was given the Nobel Prize. But I would say AI is going to penetrate almost every branch of science. But I think scientists can use it as a tool to accelerate discovery. In other words, let's say, let's talk about structure of proteins. In the old days, you had to crystallize a protein and do X-ray crystallography and solve its structure. The first protein structure took 20 years to solve, and resulted in a Nobel Prize for those, the Max Perutz and John Kendrew. And then, it became easier to do, but still you needed crystals. And typically it would take a while, at least a few months, maybe a year to solve a protein structure. Most of the time would be to try to get crystals, but it would still take quite a long time, despite all of the progress, right? Now, you can give it the sequence and to the computer, and AlphaFold will say, okay, this is the structure. So you have dramatically increased the rate at which you can get structural information. But you have to ask, why are you doing the structure? Okay, the idea is not just to solve the structure. It is to try to understand how this protein works. What is it doing in the cell? How is it working, etc. And so, if that's the goal, then the AI has made your progress faster. Okay, so you can think of it in the future as a tool that will help you, progress faster in whatever your science is.
What do you think about science that is developed in countries like Mexico and in other languages, as we were speaking also earlier? Why is it important for scientists here to develop this kind of science, even if it doesn't solve problems that are pressing of our countries?
I think this is a general problem, because science, even for much of the last few hundred years, ever since the Enlightenment and Industrial Revolution, science has really been concentrated in a few regions. For example, you could think modern science began in Europe in some sense, but very quickly science was not very strong in Southern Europe, even though Galileo was from Italy, but then Italian science was not, did not keep up. And this had to do with the politics of Europe, and the Reformation and so on. But modern science then really advanced in Northern Europe. And then in the 1900s, probably around 1920 or 30, more like 1930s, when the Nazis took over Germany and Europe started being problematic, the focus of science started moving to the United States, okay? And then the US became a very strong country. And so, even in the US, it's not evenly distributed. So there's, most of the really, if you look at the Nobel Prizes in the US, if you draw a map of the US, they're all in the Northeast or in California, okay? Maybe a few in Seattle or something like that. Or maybe one or two in Chicago, a few in Chicago. So, it's not distributed evenly throughout the US, okay? So this is a problem, and it's a problem. I often get asked this question when I visit India, like, how should India compete, in a world like this? Well, one problem is getting enough resources, okay? That's a big problem, because countries like the US or Germany or the UK, they put in a lot of resources, or Switzerland, put in a huge amount of resources into science. And, and by the way, this doesn't have to be static, because it moved from Europe to the United States, and we are in the beginning of a transition where it's moving to China or Japan or South Korea. Okay? Why is that? Because those countries are putting a lot of money into science, and a lot of money into science education and training. And many of them were trained in the US or Europe and came back. Okay? Just like many Americans went to Germany or Britain and came back. Even molecular biology, Jim Watson went to Cambridge and then came back to the US. So, and similarly, many Indians have been trained in the West, many Mexicans have been trained also in the US or in Europe. So the question is, what do they do when they come back? And the problem is it's difficult, because they don't have the same resources, they don't also have the same critical environment.
They don't even come back.
Many don't come back, like me, you know. I didn't go back to India, for example. But the problem is that it's not just the infrastructure and the resources, because there are places in Mexico which have excellent facilities. I've been to some of the institutes on one of my previous visits. And similarly, in India, there are now institutes which have very good equipment and good labs and so on. But it's also a question of, environment, intellectual environment. And one reason that, people who go to Harvard or Berkeley or the MRC where I work, is that they have senior scientists who are very role models and provide good mentorship. Because in science there's a problem. If you choose a problem that you think is very important, it may be too difficult, and this is not the time to do it. Or if you do it, maybe you're not using the right approach. Maybe you should be using a different approach. So that's where, mentorship is important, where people can guide you and say, you should wait to do this problem, you should do this other problem first, and then you can tackle this. Or you should be using this other approach. Or they can even say they have a lot of networks. So they can say, I was talking to my friend at a meeting, and he had this idea that this kind of approach would work for your problem. So you get this kind of, constant feedback. And that's very difficult to create. But, China is now creating it, okay? And Japan created it some 20 or 30 years ago. So I would say it requires some dedicated people to really set high standards and bring up the standards overall.
What would you say are the doors that have been opened in science and in technology after the description of the ribosome structure? I would say the impact has still been mostly academic, okay? The ribosome was discovered in the 1950s, late 1950s, and by the 1970s, we already had a pretty good idea of what the ribosome does, that it binds mRNA. We even knew which subunit binds mRNA and which subunit makes the protein, and roughly how it works. But what we didn't know is what is the detailed mechanism by how it works. And we didn't know how the ribosome is regulated, okay? So once you know the structure, you can ask much more sophisticated questions about how the ribosome works. How does it know where to start? How does it move? How does it use energy to make that movement? What determines the accuracy? How does it know when to stop? How is it regulated? How do viruses hijack the ribosome to make its own proteins? So these are all big questions. And how's a ribosome assembled? It's a big, complicated complex. The human ribosome has more than 80 different components. And so how's the ribosome assembled? So all of these are questions we can start tackling now. And then in terms of application, one application that's current is antibiotic design. So we can figure out how antibiotics bind to the ribosome, and that might allow us to design better antibiotics. Although that is more of a business problem than a scientific problem. Financially, companies don't want to invest in antibiotic development because they don't see as much profit in it. And then in the future, knowing the structure of the ribosome might allow you to engineer ribosomes that make different kinds of polymers. So not just natural protein polymers, but maybe different types of polymers which you don't find in nature that have useful properties. You mentioned you're working on a couple of endeavors yourself outside of academia, or can you tell us a little bit about it?
Well, one aspect obviously is writing books for the general public about the different things. So my first book was about the race for the structure of the ribosome, and my second book is Why We Age. It's called Why We Die, but it's really about the biology of aging. So that's one aspect. Another is that I'm a consultant for, I'm on the advisory board of an AI company that is trying to use AI to design drugs, to discover, useful drugs that, for various diseases. And then I'm also on a science advisor for a company that invests in biotech. Well, it invests in everything, but my particular expertise would be in biotech. And then I myself helped to found a company which is involved using mRNA for therapeutics. Sort of like the vaccines, except this is a circular RNA so that it's more stable and can be used for a much broader range of potential uses. So that's another thing that I'm doing.
That sounds very exciting.
Yes, although I'm hoping, I retired from my academic job at the end of October, so just over a month ago. But I'm hoping that all these activities about, especially this company, I hope that the company will succeed. But whether it succeeds or not, I hope it will happen in the next year or two. Because after that, I want to slowly, do other things. Like, I want to maybe read about things that interest me and just be engaged in things that I want to learn rather than,
I don't want to spoil the book because we have the editor here and he will not forgive me. But what would you say to all these people in our generation that have an urge to stop aging and to live forever?
Well, I think living forever is a mirage. It's not going to, there's never going to be a time when you will live forever. Even if they can slow down the biology of aging, you will then die of other causes, okay? I mean, if you engineered a rat to live very, very long, and you put it in the wild, it would not live very long because it would be eaten by a predator or something like that. So if you don't age, you will die of a disease or accident or violence, or there'll be hundreds of other reasons, or climate change. There are many other things that will get you. So I don't think there's this kind of immortality is realistic. But some people are asking whether you can extend life to longer. And in a way, that also is a mirage. We already live twice as long as we used to live. 150 years ago, the average life expectancy was, 40 years. And now, in many developed countries, it's close to 80 years, okay? And so the fact that we live twice as long doesn't, we're living almost 2 lifetimes, okay? If you consider 150 years ago. But it doesn't mean that, we have an extra lifetime, we should be very happy. Now we're saying, okay, we live 80, why can't we live 100? And if you live to 100, you will ask, why can't we live to 150? So this is sort of a, endless quest, I would say.
What would you do if you could live 150 years?
I don't know. I think, I think part of the reason that we're driven to do things is because we know that our lifetime is finite, okay? And so we think, we better get this stuff done, because we only have this one life to live. I think if you live, 150 years, then, there's a famous saying, mañana. You just say, okay, I don't need to do it today, I have a lot of time.
Perfect for Mexicans, actually.
So I'm not sure that we will accomplish much more. And also, I think, people are creative when they're young. And this is not only because their brain is young, it's because they are seeing life for the first time. They don't have the prejudices that you have accumulated with age. So you're more open-minded. And so many big scientific discoveries and science and mathematics have been made by people when they're young. Even in biology, it's relatively young, okay, in their 40s or so. And in other fields like mathematics or theoretical physics, it's even in their 20s, okay? So I think this is and I used to think it's only for science and mathematics, but Kazuo Ishiguro, the novelist, pointed out that this was true even in literature, that people often wrote their best novel when they were young. Like Tolstoy wrote War and Peace when he was in his 30s, okay? He was not an old man full of wisdom. It was when he was in his 30s. So I think if you lived a very long time, you might, even if your brain is still healthy, which is another big question. Can you keep the body healthy but also prevent neurodegenerative diseases? Because what we're finding now is as people live longer, the incidence of Alzheimer's and Parkinson's is growing everywhere, because the brain starts to age, even if the body is still doing okay. And so I just don't know, it is possible that I'm pessimistic and that we will solve the problem, we will have healthy brains, and we will solve the problem of open-mindedness by changing fields every 20 years. You go to a new field and you will be open-minded. Maybe it's possible, but I frankly doubt it.
You know how us Mexicans, we have a very special and intense relationship with death. And we believe that we'll live forever as long as people remember us in our families. And they put their altars and our picture and the food that we like. And I know it's very difficult that we forget you because you are a Nobel laureate, but what would you want for humanity to remember about you? For you to be forever?
I almost, at some level I don't care because I'm dead. Maybe hopefully my children and grandchildren remember me as a nice guy. But, other than that, it's not that important. And I know this is an obsession, like rich people, or pharaohs in Egypt, they all want to be remembered. They're terribly worried about being forgotten. And so they build monuments or they, create, all sorts of things so that people will remember them. And there is this idea, to go back, people deal with death in 4 different ways. One of them is called Plan A. Plan A is to simply try not to die, to stay alive, okay? Plan B is many religions, like Abrahamic religions, believe that your whole body will be resurrected on Judgment Day, and then you will go to heaven, but with your body. And that's why, Christians, Jews, and Muslims, they believe in burial. They do not believe in cremation, traditionally, okay? Because the body has to be intact so it can come alive. Then Eastern religions say, okay, your body may die, but you have a soul that can be immortal and will occupy some other body, or eventually it can escape and be by itself. So that's Plan C, okay? And Plan D is to live on through monuments or your works, okay? So if you're very rich, you can give money to have a building named after you, or you can build pyramids like the pharaohs or things like that, okay? And so then you are immortal through your monuments. Or if you don't have money but you have a lot of creativity, you can be like Beethoven or Einstein, and you can be remembered through your works, okay? But Woody Allen, I don't have to tell you who he is. Woody Allen said, I don't want to live on, in the hearts of my countrymen through my works. I want to live on in my apartment. Okay? And so this goes back to plan A. Everybody really wants plan A. The other things are only because they are worried that plan A is not going to work.
So if you want to know like the deep reason, the molecular reason of why we die, we can read the book in detail. Doctor, so we're going to open up the mic for people in the audience and also the ones that are following through the stream, ask you some questions. So there's an opportunity for you to ask questions to Professor Ramakrishnan. So we have one first question. So if you can ask your questions in English, that would be better. Otherwise, I will translate for Professor, if he doesn't understand, because he speaks fairly good Spanish.
Welcome to the Polytechnic Institute, Mexico. Thank you for sharing all of these things. We teach molecular biology for several decades now. I teach molecular biology. And I have a difficulty explaining how the ribosome was formed in the first place. What are your thoughts on that?
It's a fantastic question for which there is no good answer. Okay? And the paradox is that the ribosome is the big molecular machine that is used to make proteins by reading the genetic code. But the ribosome itself is made of proteins, okay? So, first of all, before you had the ribosome, why would you even need proteins? Obviously, life was existing without proteins. And secondly, if the ribosome is made of proteins, how did the ribosome even, form in the first place? So the first answer to the paradox was thought of by Francis Crick. And he said that maybe the first ribosomes had no proteins and only consisted of RNA, okay? And people read it, but didn't comment much about it, because when Crick proposed that, RNA was not thought of as being active chemically. So they could not imagine that RNA would be able to carry out all of the reactions of the ribosome, like joining the peptide bond and moving and so on. They thought of it as an inert molecule. But then RNA catalysis was discovered, and this led to the idea of the RNA world, that life began with RNA, and DNA even came later. Life began with RNA because RNA could both carry genetic information, but it could also carry out chemical reactions. And the ribosome may have started initially as a way of making just random peptides, which proved to be useful. And then slowly, and that may have started with the large subunit, and slowly it became advantageous to bring two amino acids together. That may have been the beginning of tRNA, and then mRNA to attach to it. And then slowly a genetic code could develop, and a small subunit along with that could develop. So it's very complicated. This is only the broad outline, but nobody really has an idea exactly how this took place.
Maybe your students could answer that question someday. Another question?
Thank you very much for being here. Different people say about the next pandemics may be caused by superbacteria, highly resistant to all type of different antibiotics. And one of the specific targets by excellence is the ribosomes for different antibiotics to be developed. So what about now? Do you think, what is the answer about structural biology or ribosomes? Do ribosome has an answer about the next pandemic coming from superbacteria?
Well, I mean, ribosome is one of the targets. I mean, it's a very common target that is naturally chosen because there are many antibiotics that act on the ribosome. And it is possible to try to use the structure to engineer better antibiotics. The problem is that, as I mentioned before, antibiotic development is not favored by big pharmaceutical companies, okay? They would rather, their favorite would be chronic diseases, where you have to take a pill every single day, so you're a good customer, okay? But if you develop a new antibiotic, first of all, it will be reserved only for resistant bacteria, because you don't want to use it unnecessarily. So the patient pool is small, and secondly, it's limited, because if the antibiotic is good, the patient is cured in a week or two, and then they're no longer a customer, right? So the profit motive, it doesn't work well for antibiotics today. And another problem is a lot of the development is done in the first world, and infectious disease are more of a problem in poorer countries. So I think the problem is more economic and political than it is scientific. Because now there are powerful tools for trying to develop antibiotics against many targets. And the other problem is that just with the COVID pandemic, we don't do things in advance to prepare for things. We wait until it becomes a serious problem, then we react, okay? And I'm afraid we have not learned the lesson from the COVID pandemic, that we should be ready for future pandemics. And in fact, the world is becoming more interconnected. So in the old days, new diseases would break out and then they would die out because people were not moving around. But now people are moving around, and so you can see, a virus that originated in China, within 2 months it had spread, throughout the world.
Thank you very much.
Hi again. So I have a couple of questions. The first one is, I kind of know the answer, but I would really love you to tell the young students, young people, why
I
should
hope
we
I have the right answer.
Don't worry, this is not an artist question. So, why should we continue doing basic science? That's the first question.
What's that?
Why should we continue doing basic science?
Oh, I think, well, first of all, the future is going to be highly dependent on science and technology. And I don't think any country is going to survive based on cheap labor, or on natural resources, okay? The reason for cheap labor not being as good a factor is because of automation, so cheap labor will no longer be a big issue because of robotics and automation. And natural resources historically have never been, advantageous. If you look at countries with lots of natural resources, like South America, Africa, they're not rich, okay? But countries with no natural resources, like Singapore, okay, are extremely rich now, okay? Or Switzerland has almost no natural resources, extremely rich. And because knowledge and technology is more beneficial and better for the economy than natural resources. So there's no alternative. Now, the question is, you could ask, okay, why should we invest in basic science? Let the North Americans, let the guys in the United States do the basic science, and we will take their ideas and apply them. But it doesn't work like that, because first of all, if a discovery is made in California, who do you think is going to exploit it first? It's going to be other Californians who know about it, okay? And they will start their startup company and produce things and so on, and that's why you have Google or, companies like that, Microsoft, etc. And so you need basic science, but even if you're able to apply it, you will not be able to apply it unless you have a pool of scientists who even understand the discovery in order to apply it. Okay? So, for example, recombinant DNA technology, right? Everybody can apply it. You know, you can have, I'm sure Mexico has biotech companies that are using, you know, recombinant methods and other biotech methods. But they could not do it unless there are trained Mexican scientists who are molecular biologists, who understand it, who train people who can go and work for those companies. Okay? So I think it's impossible not to invest in basic science. And even if you want to only take advantage of the applications. Now, the problem is that how much should you invest? Now, that's a political question. Okay? It's a political question everywhere, even in the US. Okay? How much should go for basic science versus targeted science, you know, like curing cancer or developing some, you know, technology, and how much should go for fundamental research? That's always a political question, but it cannot be zero. It has to be, you know, some fraction, significant fraction.
The last question is, with the upbringing of the genomics science and all the omics in the science, in my opinion, a lot of people has stopped working on the lab bench, and they rely basically on the omics. And I think that's a mistake, but I would like to hear what do you think about it.
I think that's, you know, there's no question that large-scale science in biology, for example, sequencing, you know, at a large scale, has been extremely powerful, even to understand individual problems. For example, you know, we've used deep sequencing, you know, RNA sequencing methods to identify some RNA in our structure. So I think, you know, the technology and the tools have become powerful, but really, science depends on many different types of problems. Some of them will require large-scale science, some of them will require very focused science, you know, almost classical in a sense. So I don't have a strong opinion. I just think there should be diversity, and everything should not be this sort of large-scale turnkey science.
We have another question.
Dr. Winky, welcome to National Polytechnic Institute. We are very, very happy to see you here. I would like to ask you for one recommendation for young researchers. This is difficult to be Nobel Prize in Mexico because we are so poor in order to give high resolutions at several different problems. But I would like one recommendation of you for the new researchers in order to spend all his time in order to give answers as you do it before, because it's very difficult for us give information in order to do really high-level science.
Yes, but sorry, what was the question?
The recommendation for young researchers.
I think, you know, it is a very difficult problem, because if you start comparing yourself immediately to, you know, some of the world centers of science, then you feel frustrated. There's no question about that. But I do think that by choosing problems that you think are important and simply focusing on those problems, trying to make sure you have the resources. And this is the job of the senior people in the institutions and in government, to make sure young people have the resources, you know, the chemicals, the equipment, you know, facilities to do what they need to do. I think that you can make progress, but it always depends on how, you know, dedicated is the government, you know, to the, and charities, if there are charities that support science. How dedicated are these institutions to supporting science? Okay, the reason China did not improve its science by accident, okay, it improved because the government decided that science and technology are important, and even basic science is very important. And it created institutions, it put a lot of money and equipment, but also money for people, and even bringing people back from the US, to work in China. So it required a real dedication. So I think that's what's needed, and without that, it will always be a bit of a struggle. But people have done very good work. I'm sure people have done excellent work in Mexico, but I know when I visit India, I see people doing work that would be, considered very good work, even in a good university in the US or in Europe. So I think it is possible to work as long as you have the facilities and you have the dedication.
At the beginning, you started as physics, and now you are making research in biochemistry. How did you change from one point to another?
Well, as I told you, I went back to graduate school and to learn biology. So I didn't just jump from one field to another. I had to go backwards to learn biology and take courses in biology and learn how to do experiments, and then, so it was a slow process.
Once again, thank you so much.
And now you will jump to gardening, right? Now you will jump to gardening, right?
Gardening, yeah. That's right.
So
My wife says that I will be a terrible gardener.
So I have one question from our audience in the internet, and they say, given that your work solved the structure of the ribosome with an unprecedented detail, what do you think are the most promising techniques of imaging and microscopy or quantic simulations to study the dynamics in real time of the interactions with antibiotics?
I think both, visualizing structures like the ribosome is now no longer done the way we did. Today, almost any undergraduate student who comes to summer in my lab can solve a ribosome structure using cryo-electron microscopy, which is a technique that has been around for a long time, but which really took off around 2010 or 2012. And so that has really made structural biology more powerful. Again, it's a more powerful tool to solve more difficult problems. Computation has also made big advances. And so I think this combination of structure and molecular dynamics can actually, help with trying to understand interaction of small molecules like antibiotics with the ribosome.
Another question from the audience?
Hi, doctor. Welcome to Mexico. My question is about antibiotics, specific the oxazolidinones, for example, linezolid or tedizolid, because my PhD thesis is about the oxazolidinones. What is your opinion, specific in the PTC, peptidil transfer?
Well, they have oxazolidinones like linezolid. They have a problem, which is that they also bind to the mitochondrial ribosome. So we have in all our cells, we have organelles called mitochondria. These mitochondria are remnants of bacteria. So 2 billion years ago, a bacterium entered a larger cell, and the two components decided to live together. And they slowly evolved into higher organisms like us. So all of us have remnants of bacteria inside us called mitochondria. These mitochondria have their own ribosomes, okay? But they're more like bacterial ribosomes. They're different from our main cytoplasmic ribosomes. So linezolid will kill not only bacteria, but it will also slowly kill mitochondria. So you can only give linezolid for a certain amount of time. So because mitochondrial turnover is slow, so if you give it for 2 weeks, it's okay. You don't end up killing off the mitochondria. So it has some issues, but there are other antibiotics that bind near the peptidoglycan center, like erythromycin or azithromycin, other macrolides, and that's an area where you can develop new drugs that are potentially useful.
Over there.
Yes, hola doctor, mi pregunta va más hacia la parte emocional, podríamos decirlo así. ¿Qué fue lo que a usted realmente lo motivó desde que usted era un niño en India, en la parte de estudiante? ¿Qué realmente lo motivó a seguir, a continuar, a seguir estudiando? Evidentemente, como usted lo mencionó, el premio Nobel, todos los demás premios nunca fueron como la meta, sino más bien, ¿qué fue lo que a usted le interesó, lo inspiró para llegar a ser tan grande y que ahora todos lo admiramos por su gran trabajo?
So she says she wants to go more into the emotional part of your work, and she wants to know what was your inspiration, your leitmotiv since you were a boy in India, and then to continue all the way, even though we know that the Nobel Prize was not your goal, but what was that thing that kept you going all the way?
Well, it's hard to say, but I wasn't sure when I was young whether I wanted to be. First of all, I thought maybe I would like to be a writer, but in India, parents, nobody thinks of being a writer because it's economically so risky. To be a writer, you might starve, because nothing works out. So, I thought of going into medicine or engineering, but then I got a scholarship that allowed me, but to take that scholarship, it was a very prestigious scholarship, you had to go into basic science. And at that time, some professors had come from the United States and had made a very modern curriculum in physics. So I thought physics was very interesting. I still think it's a beautiful field. And so I decided to go into physics. And then I continued in physics until I was doing my PhD and was frustrated. So then I thought, okay, biology seems really interesting, because I read all these magazine articles. But I didn't know any biology, so I went back to graduate school to learn biology and became a biologist. So I don't know, I think a lot of it was simply, okay, what is the next thing I can do, or what should I do now? I don't think there was some long-range vision, that, I'm going to do this, and then I'm going to do this, and then I'm going to, solve the rights. No, it was not like that. It was, I think you can plan, my advice to people is you can plan a few years ahead, okay? Think about where you would like to be 4 or 5 years from now and plan on that. Beyond that, you really cannot predict. It's not easy.
Okay, we have 4 more questions and then we go to the next part of the session. Thank you.
Thank you. Do you think the problem of predicting protein folding has been solved with artificial intelligence tools?
I think it has been, certainly it has worked extremely well. I think there are cases where it may not work, and there are lots of proteins which are not structured unless they're complexed with something else. Those are parts which still cannot be predicted because the isolated protein has no structure, so there's no structure to predict, but it acquires a specific conformation, even if it's not globular, maybe extended, upon binding something else, like its target. And about 30% of the proteome consists of these intrinsically unstructured domains. So I think I would say AlphaFold is a huge advance in terms of, even if you feel you have to verify it, it's one thing to have a structure and then you can make predictions and see if that structure is correct, okay? And, it has an extremely high success rate. So I think it's a great tool. but it's not going to remove experimentation.
Over there, we have another question.
Hi, doctor. It's a real pleasure for me to be here, and I really admire you.
Sorry, where are you?
Can you stand up, please?
No, it's okay.
Okay, my question is about what do you consider the most promising discovery in aging over the past maybe 10 years?
Most promising discovery in imaging?
In aging.
In aging? Oh, in aging. Okay, well, there are many, I would say there are 3 or 4 things. One is drugs that mimic caloric restriction. So caloric restriction has been known to extend lifespan and health span as well. And there are drugs that mimic caloric restriction like rapamycin. Rapamycin has some issues because it's an immunosuppressing drug, but maybe they can use that idea to develop safer drugs or safer doses or something. That's one. The other area is compounds that specifically attack senescent cells, which tend to accumulate in old age, and reducing the number of senescent cells is shown to be beneficial in animals. And so that's another promising area. A third promising area is to see if there are factors in blood which change with age and identify factors that are useful. For example, they may be activating stem cells or preventing detrimental effects. So those are some areas. Probably the most interesting area is reprogramming of cells. And this involves using factors like Yamanaka factors to make cells reverse in development, but maybe only a little bit so that they still maintain their identity. And so that's shown to be useful, but again, applying them to humans in an effective way is going to be fairly challenging. So these are some areas. And then there's a whole area of supplementation, like NAD supplements. And I'm not sure how useful they'll be, although there are some indications that as we get older, our ability to make important biochemicals declines, and if we can supplement with precursors, we can sort of restore our metabolism to a more optimal state. So there are a number of approaches which are interesting.
Here, over here.
Okay, hello doctor, thank you so much for your time. It's an honor to have you here. Yeah. Okay, my question is, what advice would you give to young scientists who are considering moving between different disciplines to solve complex problems?
I think the main advice I always say is you have to really be interested in the problem. You have to be interested in the field, you have to be excited by the problem, and that will give you the motivation to work on it. Because as you know, going to the lab every day, it's hard work. Okay, and sometimes it's boring. It's the same work every day, day after day. You have to come in, run gels, or purify things, or do assays. That's in biology, and I'm sure in other fields it's similar. There's a lot of very routine work to be done. And the reason to do it is if you're interested in the problem. Right? And so I would say, to pick a problem, an interesting area, and interesting problem for you, and focus on that.
Thank you. I think this is our last question, right?
Hello, doctor. Nice to meet you. Sorry, I'm a little bit nervous, so I had to write it down. This is also like an emotional question. How hard was to decide to move to another country? when I think you had your whole family in India, and also what were the main challenges you had to get through, and how did you find this motivation? How did you inspire yourself every day? Because I know it was challenging moving to India to the United States.
Yeah, so I think leaving India for the United States, it was no problem at all. Okay? I was very excited to go. I was 19 years old, and I was going to graduate school, and I thought, I had seen all these Hollywood movies, and I thought, okay, I'm going, So that was no problem. Okay? And I had gone to an English school. English was essentially my mother tongue, and I was very familiar with American culture, so I had zero problem moving there. But then I had to move all over the United States, from Ohio to California to New Haven, to Long Island and then Utah. So that was difficult, because each time you have to leave your friends and leave your thing, and then you have to go to a new place where nobody knows you, and you have to then prove yourself again. And then I would say the most difficult move was to move from the United States to England, because I had to take a 50% salary cut, and I had to leave, leaving your parents is maybe not so difficult, but leaving your children is more difficult, okay? So we had 2 grown children who were in their 20s, and they had no reason, they were Americans, they were born and raised in the US, no connection to Britain, so why would they move? They were not even allowed to move. They wouldn't get a visa to move. And also they have no desire to move. So we were leaving them, and just my wife and I were going to England. So that was hard. But that was because I felt that this institute, where I work, is a very famous institute where the structure of DNA, the structure of the first proteins, the structure of the first virus, DNA sequencing, monoclonal antibodies, all of these things were done in the institute where I work. And so I felt that was a good place to go and tackle a hard problem. And it was a once-in-a-lifetime opportunity, because if I didn't do it, then other people would do it, and then I would not be talking to you today. So that was the motivation to go to England. There was the feeling that this was a once-in-a-lifetime opportunity, and money was okay. I had enough to eat and do my hobbies and so on. So it's not that I could put up with that. So the other question you ask is, how do you put up with difficulties of doing science every day? Things are not working, and so on. There are 2 things. One is you have to keep motivated by asking, why are you doing this? And you have to still be interested in the question, that you really want to know the answer. And you're willing to try many different approaches, and try to get experiments to work, correct mistakes, try other different types of experiments, because you're interested in the question. Okay? So that's very important. The other important thing is that even if you're motivated that way, it's still, you risk burning out if you're working all the time and things are not working, etc. So you have to have a social life and hobbies, other interests.
What were your hobbies at the time, for example? What were your hobbies at the time?
So, for example, I like hiking and bicycling and movies and literature and music and travel. So, you can't do all of them, but you can certainly go on bike rides on the weekend, and you can, every day, if you go for a run, sometimes, that's relieves your stress if you exercise, and also relaxes you, then you start thinking again about the problem. And you have to sleep well. Sometimes after sleeping well, you have new ideas, that's, in fact, there's a physiology behind that, which is explained in this book called Why We Sleep.
Muchas gracias.
Okay, so the very, very last question, please.
Yeah, so in your opinion, I'm here. So in your opinion, what is the next big discovery concerning the ribosome?
Well, it's hard to say because there are many areas of the ribosome that are interesting. One is a field that we have worked on for the last 10 years, but now that I'm retiring, other people are going to continue, which is how does the ribosome initiate on mRNA, and how is that process regulated, and how is that process modified by viruses? Okay, so that's one very important question. Another is, how does the ribosome, how is quality control of translation regulated? You know, so what are the mechanisms for quality control? So for example, if there's stress in the cell, this ribosomes are stalled, how is that rescued? So that's another very important question. And then ribosome assembly is an important question, which is, you know, how's the ribosome built up from all these components when the cell is divided?
Okay, so Professor Ramakrishnan, thank you so much for this chat. We really appreciate it. Right now we want to as you can see, we have passionate students and professors here that are really working towards science here in Mexico. So right now we want to give you a small symbolic gift from our Director of Outreach here in the National Institute. And also, if you could please sign books for the people who ask questions, it would be an honor if you could do this. So thank you.
Professor Benke, thank you very much for being here. It's a small present from our part. And thank you, of course, to the ESIQIE that received us, the crew of ESIQIE, of course, Tomás, Ana Cristina. Thank you very much for all the hard work to get Professor Benke here in Politécnico. Thank you very much, and thank all of you to be here today. And well, Professor Benke, if you allow us, we will make our song, our institutional song, that is called Vuelo. It is dedicated to you. So, si me ayudan a ponerse de pie para hacer un vuelo. Vamos a hacer uno primero al Politécnico y después para el doctor Benke. Va, 1, 2, 3.
Vuelo, vuelo, gloria, la cachi cachi porra, la cachi cachi porra, ping pong porra, Politécnico, Politécnico, gloria.
Y ahora para el profesor Benke. Así como está el profesor Benke. 1, 2, 3.
Vuelo, vuelo, gloria, la cachi cachi porra, ping pong porra, profesor Benke, gloria.
Thank you very much, profesor. Welcome. Muchísimas gracias a todos y a todas. Gracias a ESIQIE por recibirnos, a su director, a su equipo. Muchas gracias por recibirnos.